This lecture is part 1 of 5, covering chapter 3, titled The Chemical Basis of Life, part 2. In chapter 2 that you just looked at, you learned about atoms, how these atoms come together to form molecules and compounds, and then you also learned about the importance of water. This chapter, we're still looking at chemistry. We're going to look at organic chemistry, so kind of the molecules that are used to create the cells. In our 12 principles of biology that we went over in chapter 1, we're really going to focus on number 2, how organisms use energy, how they get this energy from these chemical molecules. We'll also look at principle number 9, how structure, specifically molecular structure, determines function of the different molecules. Number 10, how new properties come from complexity. And also number 12, how biology affects our society. So I'm going to try to relate this organic chemistry chapter to real life examples. Also, if you remember, we have our 10 levels of organization for living things. And just to point out again, we're still looking at the very small level, so we're looking at atoms just a little bit. We looked at how atoms form molecules. Now we're really going to focus on the macromolecule part. And again, we have to look at chemistry in order to understand how cells work. So we're again looking at this question, how is biology just applied chemistry? And again, you're probably like, why? Why do we have to learn about chemistry? And that's because these molecules are what are going to be put together to make our cells, that smallest living unit that we'll get to in chapter 4. So biology is founded on these principles of chemistry, like I've mentioned. And specifically, chapter 3 is looking at organic chemistry. So now we're going to see atoms and molecules that contain carbon rings and carbon chains. And these organic molecules, like I said, contain carbon. A lot of times these molecules are very, very large and complex. So we actually call them macromolecules. So macro means large, very large molecules in this chapter. And in order to understand these macromolecules, we're going to review carbon a little bit. So carbon, if you look at the periodic table, carbon has an atomic number of 6. That means carbon has 6 protons. It also has 6 neutrons in the nucleus. In addition, you're also going to have 6 electrons. And if you remember from chapter 2, the first 2 electrons go into the first shell. Then your next 4 electrons are going to go into the second shell. And those 4 electrons in that very outer shell, those are called the valence electrons. So these valence electrons, they can interact with other atoms. And remember that in order for the second shell to be happy, you want a total of 8 electrons in that outer shell. So it's already has 4, so we need 4 more electrons to make this atom really happy. This means that one carbon atom can make up to 4 bonds. These bonds are usually some type of covalent bond, so strong bonds, and they can either be single bonds or double bonds. So your carbon atom can share 1 electron, or it can share 2 electrons with another atom. I just mentioned that carbon forms covalent bonds, and it can form either the nonpolar or the polar covalent bonds, depending on what type of atom it's going to bond with. And just to review, molecules that have nonpolar bonds are blank. So how do these molecules interact with water? And I'll let you think about it. Okay, so molecules with nonpolar bonds, remember, these molecules are going to be hydrophobic, so they're going to be water-fearing. Molecules that have polar bonds, how are they going to interact with water? These molecules with polar bonds are going to be hydrophilic, so they're going to be water-loving. So some of the macromolecules we're going to look at, they're either hydrophilic, water-loving, or they can be hydrophobic, which is water-fearing. Some of the other molecules we're going to look at are called amphipathic molecules. Those are molecules that have both hydrophilic and hydrophobic regions on them at the same time. So if you look at the molecule up on your screen, we have 3 carbons in the middle of the molecule. The first 2 carbons on the left, they're bonded to hydrogen atoms. Those carbon-hydrogen bonds, or carbon-carbon bonds, the electronegativity of carbon and hydrogen are very similar, so those are nonpolar covalent bonds. So the electrons are going to be shared equally in those bonds. If you look at the third carbon on the right, that third carbon is bonded to oxygen atoms. Oxygen has a higher electronegativity, so oxygen's very greedy compared to the carbon. Therefore, these bonds between the carbon and oxygen, they're going to be polar bonds. So your electrons are going to spend more time around the oxygens compared to the carbon that they're attached to. And that causes that side of the molecule to be hydrophilic. So it's going to have polar bonds, it's going to be hydrophilic, it's going to want to be in water. The carbon-carbon bonds and carbon-hydrogen, carbon-oxygen bonds, since they're all covalent bonds, are very stable, very strong bonds. In the molecule that you just looked at, we had carbon atoms bonded to oxygen atoms. Some of these different groups are called functional groups. So if you have a group of atoms with special chemical features that are functionally important, that's called a functional group. One functional group that's up here is called a hydroxyl. And that's just when you have a carbon atom attached to an OH group. And these OH groups, you usually find them in steroids, alcohols, carbohydrates, and a few amino acids have these hydroxyl functional groups. There's lots of different types of functional groups, so table 3.1 in your book lists some of these functional groups. Up at the top we have an amino group. So this is when you have a carbon attached to a nitrogen with 2 hydrogens. Those amino groups are found in proteins. You can also have carbonyl, you can have aldehydes, there's carboxyl groups, and then our hydroxyl group that I start off with is at the bottom on this table. More examples, so you can have methyl groups, phosphate groups. These phosphate groups, so the phosphate with the oxygens around it, we're going to see that in nucleic acids, so the DNA and RNA. We see it in ATP. You also see them in some amino acids that we're going to look at. And then other functional groups have sulfur in them, so you have sulfate and sulfhydryl functional groups. So again, these functional groups, they're specific groups of molecules that are attached to our carbon chains or carbon rings. The carbon chains and rings that we're going to be building from this carbon backbone, you can end up with molecules that are called isomers of each other. Isomers is just defined as when 2 structures have identical molecular formula, so they have the same types of atoms and same number of atoms, but how those atoms are bonded to each other is different. So you have molecules that have different structures, and that different structure causes the molecules to have different functions and characteristics. So an example of an isomer is shown at the bottom down here, and this is called a structural isomer. Structural isomers, they contain the same atoms, so same type of atoms, same number of atoms, but those bonding relationships are different. If you look at the example, we have two molecules. Each of these two molecules, they have 3 carbons, 1 oxygen, and 1, 2, 3, 4, 5, 6, 7, 8 hydrogens on them. So they have the same molecular formula, but that hydroxyl group, that OH, in isopropyl alcohol, the OH is bonded into the middle carbon, whereas compare that to the propyl alcohol on the right, the hydroxyl group, the OH group, is on an end carbon in that molecule. So these are examples of structural isomers. Other types of isomers include stereoisomers. These are molecules that have identical molecular formulas, they have identical bonding relationships, but how the molecules look in 3D or spatial positioning actually differs. So one type is called a cis-trans isomer. This is the top one in the picture. Cis means that 2 atoms are on the same side of your carbon chain or ring. So if you look at the cis-butane, the 2 hydrogens in the middle, highlighted in green, they're both facing up. So cis means the same direction. Compare that to the transbutane on the right, and you see that the 2 hydrogens in the middle, again the ones that are highlighted in green, they're now facing in opposite directions. So trans just means they're going opposite in their spatial positioning. Another type of stereoisomer is called an enantiomer. This is the example shown on the bottom. Enantiomers are mirror images of each other. So think of your hands. Your hands, they have similar construction, similar shape, but they're mirror images. So no matter how hard you try, you can't overlap them to match. They just have that mirror image going on. So in this chapter, we're going to be building what are called the macromolecules. So macromolecules, again, are large molecules. These macromolecules, we're going to build them up by using a condensation or dehydration reaction. So these reactions, we're going to link monomers, and monomers are just the building blocks of a macromolecule. So think of them as like Legos. We're going to put those monomers, those building blocks together to form polymers. Poly's many, so just many of these monomers stuck together. And as molecules go through the condensation or dehydration reaction, you're actually going to dehydrate your monomers, and you'll end up with molecules being produced as a byproduct. So that's where that condensation or dehydration name comes from. Every once in a while, a cell wants to take a polymer, a really large molecule, and break it down into its building blocks, or break it back down into the monomers. This occurs by a hydrolysis reaction. So it's basically the opposite of the dehydration reaction. So you're going to take your polymer, your really large molecule, and you're going to break it down into the monomers, those building blocks. And in order to do this, you add water, so you're going to use a water molecule to break things apart. So again, hydro's water, hydrolysis, you add water, things break apart. So I'm going to show you the condensation or dehydration reaction first. So up on your slide you have 2 monomers, which are represented by the white circles. Those 2 monomers, they're going to bond together. And as the monomers come together, you can see there's a water molecule coming out at the top. So we're dehydrating those 2 molecules. That causes the 2 monomers to bond to each other. So now we have the 2 white circles bonded. And you just keep on adding these monomers onto your growing molecule. So we can take that 2 monomer molecule we have on the right, add another monomer to it. In the middle, you dehydrate it so you get out another water molecule up at the top. And now you have a molecule that has 3 monomers attached together on the very right. And we can just do this again. We can add a 4th monomer coming in. We knock out another water molecule, and you end up with a molecule that has 4 white circles attached together. So that's your dehydration or condensation reaction. Now we're going to take that 4 monomer molecule that we have on the right, and we're going to break it down using the hydrolysis reaction. We're going to take that 4 monomer molecule that we just built, and we're going to break it down. In order to do the hydrolysis reaction, we're going to add a water molecule that's coming in at the bottom in the blue box. When you add the water molecule, it breaks off one of those monomers, one of those white circles. So we go from a molecule that's 4 monomers long to a molecule that only has 3 of these monomers attached. Then you just keep on adding water molecules. So we'll add another water molecule in, knocks off another monomer, so we get down to 2 of them attached together. And then finally you add the last water molecule, and it breaks apart the 2 final white circles at the end. So again, this is showing that hydrolysis reaction, how we start with a polymer, so a large molecule, we add water to it and break it down into its monomers, or those building blocks. And this is just summarizing. So up at the top we have that dehydration reaction, how we put monomers together to form a polymer. The bottom reaction is the hydrolysis reaction. You take your polymer, add water, and you end up with monomers from this reaction. This first part of chapter 3, we looked at organic chemistry. So remember, organic chemistry is just looking at molecules that contain carbon, rings, chains. Also contains a lot of hydrogen and oxygen in it. These carbon-based molecules, they usually have covalent bonds, and they can either have polar or non-polar covalent bonds. And this allows molecules to have polar and non-polar regions on them. And thus, because of that, they can have hydrophilic regions and also hydrophobic regions on a molecule. We looked at functional groups. Those are things like the hydroxyl, the carboxyl, the phosphate group. So those are groups of atoms that have specific functions to them. And then finally we looked at how these macromolecules are going to be synthesized or created, and then also how we can break them down.